All right. Well, Alex, you're a student of the Carnivore Certification Program, and this is another consultation sort of event. You had some questions about some of the modules that I talked about in the course, and this is your opportunity to ask me any of them, and I will, of course, to the best of my abilities, answer them. And yeah, just shoot. These are fun. These are always fun to do. So
Yeah, thanks for having me, Andy.
yep.
So I guess I want to start with the vitamin C module and I guess the related topics there. So I wanted to ask why excess vitamin C is converted into oxalates, and I guess does it have any physiological purpose?
I'm not entirely sure. I know that the reason why it turns into oxalates, there's 3 different compounds that sort of in excess turn into, or rather, sorry, excess vitamin C does not only convert into oxalates in excess. There's other alates, so to speak, that it converts to as well.
Really?
Yeah, the main one, though, is oxalates, at least at the top of my head, and it's the one of most relevance. It has to do with its chemical structure. I'm not entirely sure why. That's something that I have to look into myself. I wish I knew the answer to that. It's a good question. But I don't think it actually does serve any physiological purpose. So, for example, usually oxalates are produced in very small quantities as a metabolic byproduct of amino acid metabolism. Hydroxyproline is involved in that process. And even that, it's basically a waste product because it's urinated out. It's eliminated. And from everything we've observed, it does not seem to serve any other physiological function. Oxalates themselves, they crystallize and form raphides, and they just obliterate cells. And so it would make sense that your body could chemically make them as a byproduct of something and then sort of excrete it just like any other industrial waste product, like on a macro level in a machine realm. But that is a good question. I'll probably write that down and figure out why. I would almost guarantee it is simply just to do with its chemical structure. Probably has a higher propensity to turn into oxalates, but I don't think there's any meaning behind it. I don't know if it's meaningful, if that makes sense.
Okay.
Yeah.
I guess the idea I had in mind was like for glucose, which is also produced in the body, but it converts to fat if consumed in excess. So I guess that was sort of my mindset with vitamin C.
Yeah.
And I guess leading on to that, what factors beyond blood sugar can influence cellular redox potential?
Other than blood sugar, basically the reason why we say that hyperglycemia can cause an issue with redox potential is precisely because of the inflammation that it induces. Anything that causes inflammation is what we say reduces redox potential. And the reason for that is it's pretty intricate cellular biology, but what ends up happening when you have inflammation is in order to stimulate the activation of pro-inflammatory cytokines, you have to have it's something called phosphorylation, which requires inorganic phosphate to attach to something. But the more that you have inorganic phosphate floating the cytosol, you're going to have a commensurate increase in ADP. And so you have a slowing down of the mitochondrial oxidative capacity. And what that will do is it will induce inflammation because either you'll have something that will induce inflammation because it slows down the rate that the mitochondria function, or it's the opposite where you're supposed to undergo inflammation because you've actually injured something. Either way, the oxidative potential decreases because the NAD to NADH ratio will go down. You'll have more NADH because you can't get enough NADH to the electron transport chain. Then you have NAD positive being replenished. And so you will have a decrease in redox potential that will cause, overall cellular decrease in redox potential, meaning that your ability to sequester substrate like glucose goes down. That's why I think that inflammation can be a really big exacerbator of diabetes. If there were some way to have diabetes without inflammation, I think that your diabetes would actually be lower, less severe for that reason. But of course, that's not the world we live in. But and just as a final note, because I hope that clears that up, because it's not exactly that hyperglycemia is just one thing that can cause a lowering of redox potential. It's that anything that causes inflammation does because of that mechanism. If you're inflamed, you have a lowering of redox potential. That's what's going on. So we know that glucose can harm cell organelles above certain concentrations, so that's why we say that. But just as a final note, because you are definitely, I can already tell that you have a learning mentality. You're like me in that regard, like a student mentality. I will give you this final iota of information. simply having a decreasing redox potential that first causes inflammation. But if you decrease cellular redox potential enough, you'll actually get...
Sorry, Eddie, it cut out for a bit. So.
Oh, okay, sorry about that. Yeah, I saw that for a minute. When you have a decrease in redox potential, it'll start to cause inflammation, or really, again, or vice versa. If you get inflammation, it's a lowering of redox potential. But if it lowers enough, you actually get a complete elimination of inflammation. Your immune system shuts down in the cell. And then it goes into overdrive, like an energy crisis situation. That's when you get a buildup of AMP instead of ADP, and then it activates AMP kinase, and then you get all the anti-inflammatory pathways that are activated by that. Metformin actually activates that pathway. So,
that makes sense.
Yeah. So,
anyways, I guess I'll go into the next question now. How does, I guess, reintroducing low oxalate foods mitigate the effects of oxalate dumping to some extent? Because you usually hear people say, if you're experiencing oxalate dumping, you should eat some like low oxalate foods or vitamin C foods to try and mitigate those effects.
Yeah, I've heard this before. It definitely seems to work for people that are experiencing symptoms that seem to be a phenomenon of oxalate dumping, which, by the way, needs a lot more research done on it. Because it's something that, the medical community does not really recognize as an issue. It would also go against their ideology in many ways. That's also a good question. I've speculated about this before. Since there isn't a lot of research to begin with, like I just said, it's really hard to it's really up for speculation. It's up for hypothesizing. I believe that your body is really efficient and proficient at recognizing when to stay in sort of, I don't want to say panic mode, but like almost a crisis mode where it has to stay in a sort of, not fight or flight situation. I'm trying to think of a great word to explain it. When you're undergoing exercise, for example, there are changes that during the exercise occur in the body and they linger afterwards. And then once it realizes that the stress is gone, you sort of have a relaxation thing. It just seems to be a case of when the body recognizes that the introduction of oxalates is done, it's sort of like, looking around the corner and going, okay, I think we're good. And then they just flush out everything. It doesn't seem, it's really ironic for me to say this. It doesn't seem to be logical. But of course, the reason I say it's ironic is because, of course, the body knows what it's doing. And I'm the one to say that all the time. But it's something that I think definitely needs more research on it. Because these are questions that just delve into things that no one knows, really. Especially considering the phenomenon of oxalate dumping is pretty new in the health space. Because no one was really dumping, they weren't eliminating plant foods. The idea of eliminating plant foods is relatively new in the health space, even though it's taking traction. But yeah, it's something that I wish that I had an answer to, but it's not something that we have an answer to yet. But that's a speculation thing, so.
And I guess just to add on that, if this doesn't have an answer as well, by all means, that's okay, but why would permanent damage from oxalate dumping requires someone to consume carbohydrates again? I think you mentioned this in a few videos where you mentioned Sally K. Norton.
Yes. Okay. Now I understand what you're saying. So usually I've seen that people that report oxalate dumping, they have issues they have permanent cellular damage to sometimes their entire body. You gotta think, when people are oxalate dumping, it's from every part of their body. So it's affecting most of their cells to some extent. And one of the cells that it can target is the liver. And so the liver is responsible for producing lots of glucose. It's the sole part of glucose production. You've got the kidneys too, and then you've got the small intestine to a very small extent, and to an even smaller extent, you got the brain, a really small extent. But given that fact, I've seen that some people, when they introduce carbohydrates back in, they can have a better regulation of glucose than otherwise if they have ostensible oxalate toxicity. So, of course, that's why the general rule is still don't eat carbs, right? Because the general population is not damaged from oxalates to that severe of an extent. But some people are. And what I will say is association does not equal causation. So when people introduce carbohydrates back in, is that what is causing them to have better experiences on carnivore, the carbohydrates themselves, if they're oxalate, if they have damage from oxalates, or is it because the carbohydrates they're consuming have oxalates in them? We have to look at the food that they're actually consuming, and then, try and infer to the best of our abilities what the factor is, if it's only one, maybe it's multiple, that are actually affecting these people in a positive way. And to anyone that's actually listening to this, it's why I often say all the time in my videos, I go, hey, these people that are ameliorating their issues with carnivore, but the oxalate toxicity seems to be getting to them and they need some carbs or something, they are only utilizing like 20 grams to get to the effect that they need. It doesn't mean like go Paul Saladino and like dump hundreds of grams down your throat. It's not what it means. Here in the space, I think a lot of people can be afraid to introduce carbohydrates back in because of the dogmatic element. And I understand a lot of people might watch my channel and think that I'm one of those people because of the way that I talk. I'm not at all. I'm not like that. It's not what I do. But I understand that many people might be afraid to even introduce 10 to 20, but don't feel guilty about it, is what I would say to anyone that's worried about that. I don't demonize carbs per se. We just know that they're not necessary as a general rule, and above a certain concentration in the bloodstream, they are toxic. That is a fact. So anyway, I know that was a little tangential maybe, but hopefully it clears everything up.
No, I think that helps with like structuring everything nicely.
Yeah. Good.
All right, so I now want to go into some of the mitochondria related topics.
Oh, okay.
And so you mentioned that coupled mitochondria convert food energy into chemical energy in a 1-to-1 ratio.
Yeah.
While uncoupled use more and result in chemical inefficiencies or more heat. Could you maybe elaborate on how that 1-to-1 ratio is maintained in the coupled and how it changes with uncoupled?
Yeah, the words coupled and uncoupled refer to basically whether or not the proton gradient. So I don't know how familiar you are with the structure of mitochondria, but you've got the intermembrane space and it has a lot of protons in it, or at least that's what they say. It's protons, H+. In reality, they exist as H3O+, but it's a technical detail that really doesn't need to be emphasized too much. And there's a gradient. They often say there's a concentration gradient between H+ ions in the intermembrane space and in the matrix. And basically, the thing that makes the ATP synthesis, the ATP synthase motor, protein is really what we should say is spin, is the protons from the intermembrane space flowing through into the matrix. And then it spins the motor and it causes, ADP and PI molecules that are on the subunits, the alpha and beta subunits, to synthesize into ATP, and then the cycle continues, basically. First of all, the problem with that is believing that there's a concentration gradient between protons, because there's not, because that implies that protons are basically independent variables, which they're not. They're dependent. This is the whole thing about pH regulation. You've got 4 factors that influence pH regulation and nothing else. You cannot change the concentration of H+ ions in a solution by adding them to a solution. So it's not true that it's just a concentration gradient. But coupled refers to a coupled rate between the protons flowing into the matrix and the rate of ATP synthesis. So for every proton that flows through, you've got an ATP that's synthesized. Okay.
Right.
Uncoupled means that there are separate hydrophobic pores that are produced, usually through different cellular signaling. So hormonal signaling, if you've got a low insulin, this is when it pertains to a fat cell. You'll actually have a fat cell that will respond to low levels of insulin and high levels of glucagon by opening up a hydrophobic pore or two. I know that it's at least one, where it's completely independent. You have H+ ions that are allowed to flow through into the matrix that have no effect on the ATP synthase motor. And so you'll actually have a rate of, you'll have a higher rate of nutrient utilization to keep up with the protons flowing in, but you won't have a congruent amount of ATP that's produced. And so what happens is you get this high rate of heat that's produced because the engine is going at the same rate, but the ATP synthase motor is also going at the same rate, but it's not going at the same rate as the overall mitochondria is. or mitochondrian on a singular basis. And so you've got a lot of heat that's produced because you're still using the same amount of food. So the energy has to go somewhere. It turns into calories and then they're released. But the rate of ATP created is not the same. So let's say that cell needs more ATP. Let's say it's undergoing some process that requires more ATP. Well, then it's going to use even more food too. And I keep saying food again. I mean, we're talking about mass constituents. We're talking about like NADH and all that stuff. But, on a simplistic basis, if we can even get it to be super simplistic, it's a very complicated topic. You get this issue where it's not an issue, but you get this phenomenon where a bunch of substrate is coming in, but you're not meeting the ATP requirements. So in order to meet the ATP requirements, it uses even more. So it's just that's what we mean by uncoupled versus coupled.
Oh, okay.
Yeah, and so it happens in fat cells over longer periods of time. It turns them from white adipose tissue into, I know I said in the video, it turns them into brown adipose tissue. It turns them into, it browns them. It doesn't completely turn them brown, I don't think. It may, but I think it more so turns to browning tissue, like browned, not completely brown adipose tissue. And it only happens in fat cells because the thing is if it happened in any other cell, I mean, there was actually a drug that did this 2,4-dinitrophenol. You may have heard of that drug before, maybe. It had a lot of controversy because it did exactly that. It was known as like this miracle weight loss drug. And you could take it, and it uncoupled all of your mitochondria. But it uncoupled all of them. It didn't just do it in your fat cells. It did it in your muscle cells. It did it in liver cells. And people were... No, it's not good at all. The cataracts developed in people, children even. And it's kind of sad that children were even on these drugs in the first place. But and then people died prematurely. It was really, really bad. And then it was recalled. But that's actually why it worked. People were losing weight rapidly because of that phenomenon. So, yeah. Anyway.
Okay. Wow, that was a very heavy one.
Yeah, I know. I really, again, when you get into it's a warning. When you get into the granular details, I'm really happy to answer them. It's just a matter of, it's going to get complicated very quickly.
I just need a minute to process all of that at once. So, I might always come back to this video.
Yeah, I was going to say it's a good thing that we're recording it because then you can watch it and then be like, I can send this to you afterwards as well, even before it's uploaded. Yeah.
And now I want to go into some details regarding the Randle Cycle. This was sort of inspired because of a recent video by Dr. Abs, which I think was more focused on NAD as the main topic, but in any case, that got me interested in NAD as a whole. And so I guess I want to first ask, what exactly is the role of NAD in the Randle Cycle anyways?
In terms of, so I think that he may have been referring to redox potential, because the thing about the Randle Cycle is that whenever you're having that cross inhibition happening between fatty acids and glucose, there's this phenomenon that, if we're going to be extremely technical, it is technically a theory, yes, but it's only because the research that's been done on the Randle Cycle is very, very few and far between. As much as there is for someone that hasn't looked at the medical literature a lot, it seems like it's a lot comparatively to all the other things that have been researched. It isn't that much. So what the hypothesis, what the theory is that when you have this cross inhibition of fatty acids and glucose, you get a decrease in redox potential because, let's again, trying to make it not so complicated, it's almost impossible. Let's start from the very beginning. Let's say that you're oxidizing a lot of fat. Fatty acids will enter a cell passively, and they enter it through a CD36 transporter. They go through, and then they go to the mitochondria, because in order for them to even be used to create ATP, they have to go in the mitochondria. It's an exclusively mitochondrial process. So they enter through CPT1. And you're producing a lot of acetyl-CoA that way. You're producing a lot of ATP. Great. Okay. Now imagine that you dump a bunch of glucose in, and you've got the situation where glucose, GLUT4, you release insulin, GLUT4 is stimulated to the plasma membrane, and then you've got glucose coming in, and that's going through a separate pathway. And then you create a bunch of pyruvate. And pyruvate goes through the mitochondria, but it can't be used to produce the things it's trying to produce. It can't be used to create acetyl-CoA because the acetyl-CoA that's building up from fat is blocking the pyruvate decarboxylase, or pyruvate dehydrogenase rather, enzyme. And so it leads into this other thing, whatever. I'm trying to figure out a good way of explaining this. A buildup of acetyl-CoA in the mitochondria will migrate to the cytosol, and it will inhibit a lot of the glycolysis enzymes. So if you can imagine those graphics, you'll actually see that acetyl-CoA turns into citrate, really, and then citrate migrates out, and it blocks a bunch of transporters. What that means, though, is that if you've got a block between two pathways, and they hit a sort of gridlock where not one of them can even get into the mitochondria at all, it slows the rate that substrate can even reach the mitochondria, which then subsequently causes a decrease in acetyl-CoA concentrations. So first you have to have an increase, then it blocks the enzymes, and then it causes them to neither one of them to be able to reach the mitochondria at the rate that they're supposed to, that they otherwise would, which causes a decrease in acetyl-CoA. In fact, the best way of actually imagining this is taking both of the graphics, the one where the acetyl-CoA, or really citrate, is blocking one of the pathways, and then then the other one where it's blocking the other, and actually overlapping them. You can
overlapping.
Yeah, basically what you could do is not literally overlapping the graphics, but you can. I did this myself actually. I took a drawing. I drew one of them out, and then the other one I drew an inhibition line. You know, the ones that look like T's. You know, I drew them, the ones that weren't included in the first graphic. And you could see that all of them were blocked. CPT1 was blocked, so fatty acids couldn't get in the cell. But so was GLUT4, and so was PFK1 and PFK2, and so was one that's not even depicted is pyruvate kinase. That one isn't even depicted, which is that's the enzyme that takes enolase and converts it into pyruvate. So you actually aren't even producing a lot of pyruvate. Even if you were, it wouldn't be able to get in the mitochondria. But it inhibits so many different things. And you realize, oh, well, if you have an increase in acetyl-CoA because one of the pathways is producing a lot of acetyl-CoA, you have an inhibition of the other, which explains why one of them isn't going to be able to get in until the other one is used up. But if you have both of them coming in at the same time, because you've eaten a mixed meal and you have mixed hormonal signaling, you're going to have a gridlock situation where they're like both sticking up against each other, like friction almost, and they can't get to the mitochondria as fast as they otherwise would. And so, sure, the thing that initiates the Randle cycle is a buildup of acetyl-CoA, but eventually that subsequently is followed by a decrease in acetyl-CoA. So the whole reason I just brought this up is because you brought up NAD. When you have a decrease in the redox potential because you've decreased the rate that the mitochondria are functioning, you're going to have a decrease in NAD positive. And I think if he was talking about NAD positive, he probably was talking about how the Randle cycle will cause a decrease in NAD positive, at least acutely, whenever you've consumed the meal. And then you could have a feedback loop where that causes you to not be able to oxidize glucose as proficiently, and then that causes a decrease in redox potential because of glycation, and then it's just this whole messed up thing. So I'd have to hear exactly what he said, but I know that he promotes the sale of NAD positive, which has a very important critical role to play. I'd like to talk to Dr. Abs personally about whether he would rather sell the supplement or get people to optimize their NAD positive to NADH ratio with diet.
I believe he went into detail on trying to optimize it with diet primarily because many physicians have been inappropriately prescribing NAD and
right,
be hospitalized and such. So. Right.
I mean, you got to think, if your ratio of NAD to NADH is impaired because you have a decrease in mitochondrial activity, the only thing I can describe it as is palliative. It's counterproductive as well to give someone high NAD. You're trying to increase the numerator without decreasing the denominator. You know what I mean? It's not a great way of doing it. I mean, it's like, well, you're not fixing the root issue. Like, okay, my ratio theoretically, it's 1 to 4, which obviously that's not. You've got like 700 to something else with NAD positive. I'm just using a good, an adequate, simplistic ratio. My ratio is 1 to 4. That's too low. I want it higher. Well, instead of making it 1/3 or 1/2, instead I'm trying to make it 2/4, 3/4 without actually fixing the issue. And it's like, that's not, I don't think that's a good approach. I understand why it can be a transitional thing. When people aren't in adequate health and they haven't fixed their diet yet, if they're in the middle of doing it, they can take those as well. I've actually said that about Cerule. I don't recommend people do that at the same time unless, what I usually say is, okay, fix your diet first. If you want some assistance, you can take Cerule, but don't feel obligated. And there you go. So anyway, maybe that's how he's promoting NAD positive. Not sure, but.
And I guess, this will probably be the last question. And
okay.
I'm not sure if there's much you can really discuss with this, but I was curious, do artificial sweeteners have any impact on how the Randle cycle would be upregulated and such? Like, would it maybe affect protein turnover or something, even if you were to not consume one substrate or another?
That's a good question that I haven't even thought about. Now I'm thinking about it, and I think that you probably have a point. I'd have to think more about the protein turnover thing, but the first thing that actually comes to mind is something that Ben Bickman talked about in actually a video that I critiqued, ironically enough, of his, and it was actually on the Randle cycle, and he talked about how the role of hormones with the Randle cycle. And he's right to mention that there's a role that these play. I mean, we know that it's not just about allosteric stimulators and inhibitors like citrate and stuff inhibiting enzymes. Hormones are overarching regulators of systems. And what he made sure to mention was that insulin also influences the rate at which glycolysis is occurring versus beta oxidation. So in terms of what the cell will actually use, he said it depends on hormones. That's why I critiqued it, because it was like, well, they're not dictators. I mean, you can have high insulin, but insulin resistance exists, right? So it's like you can't say that they're dictators of fuel use if one of them elevated doesn't cause glycolysis to ensue or something. But that does mean that since they have influence over which pathway is being used, and therefore how much of a decrease in redox potential results, because of course, the higher the redox potential, the more the redox potential lowers is usually commensurate with a 50-50, like if you've got 50% of your substrate being 50% of the cell oxidizing glucose and 50% of the cell oxidizing fat, you've got the highest rate of gridlock because they're both competing at the same rate. Versus if you had 90-10 or something. So since insulin influences that ratio as well as glucagon, it is a fair assertion to say that anything that affects the I to G ratio, like sweeteners, can have an effect on the Randle cycle. However, if it's in the context of someone consuming a diet that isn't mixed, like it isn't mixed with macronutrients, let's say with us carnivores, we're oxidizing mainly fat. I think it can have an effect on the rate of glycolysis, or the influence on a cell to undertake glycolysis at least. But the ramifications are not going to be as severe as if you were eating a mixed meal. Yeah. So that's what I would say. But you do have a point. You do raise a point. And you also raise a point with even the production of glucose. I mean, any substrate that's involved in the Randle cycle, so glucose, fat, then you've got insulin and glucagon and all that stuff. If a sweetener can affect any four of those things, then it can theoretically have an effect on the gridlock, so to speak. So insulin is a regulator of gluconeogenesis, as we know. It's not a complete regulator. I've seen studies that show that hyperinsulinemia only inhibits gluconeogenesis by 20%. However, then you have to look at the people that were studied. And then you have to look at, what does hyperinsulinemia mean? What was the level? How long was it? It's complicated, but it's not a complete regulator, but it is a regulator. And so it can also inhibit the rate of glucose production because, first of all, it inhibits the rate, but also insulin is going to have sort of at least an influence on ushering glucose out of the bloodstream that's already in it. And so that's another reason why people will say, well, if you drink a bunch of sweeteners, you may become more hungry than otherwise. Because what happens when your glucose lowers, you're going to get hungry. It's going to stimulate your appetite. So yes, I know that's a very long-winded response to your question, but yes, I do believe that is a good point. It probably does, by my estimation. Of course, with any questions like that are that intricate, you've got to perform a study or something like that, in vivo kind of thing. But I do like talking about these topics because it's one of the things. I mean, it's what I've made my name known for on my channel, is getting into the chemistry of the body. So, yeah, that's good. If you've got any more, I know that you said that was maybe the last one, but if you got any more, I'll be happy to answer because I got time.
Yeah. I don't know.
I don't want to pressure you into asking any more if you don't have any. But you know,
perhaps just one thing which it was a little iffy for me when you first explained it in the module, but I guess I want to touch on the how heart disease occurs, or more so like when you went into the idea of reduced shear stress contributing to hypertension, I believe it was. Could you maybe elaborate a bit further on that? I don't know. It just didn't come into me as clearly as I wanted it to.
Yeah, so basically this is a distinction between turbulent blood flow and shear stress, basically, the shearing force of blood. So the whole point of me making that distinction was because people, including me, are most likely, they're led to believe that on the surface, if they were given two images, one was of turbulent blood flow, which involves eddying and pools of blood in, an area that has a bifurcation. So you've got like 3 different arteries meeting at one sect. If they were to see an image of that, and then they were to be shown an image of blood piercing at a very concentrated source of the artery. Did I cut out? Sorry, I don't.
Yeah, sorry, you cut out.
Okay, basically, if you've got an image of turbulent blood flow that they're looking at, and then they're provided with an image of a shearing force of blood hitting a concentrated point in the artery. Most people, again, including me, are often led to believe that shearing force, the shear stress, is what would be most conducive to developing plaque in that area, as opposed to the turbulent flow. When in reality, paradoxically, that's not the case. Our body seems to be far more adept at withstanding shear stress on a regular basis than it is at basically withstanding exacerbated turbulent blood flow. And it's because, and so you'll get this swirling and editing of blood that is exacerbated by hypertension. And the causes of hypertension, I feel like the major cause of it is inflammation, where you've got inflammation in other parts of your body, which involves vasodilation. But in order to compensate for the higher demand of blood, you have vasoconstriction in the central part of the heart, of the cardiac system, which can increase blood pressure. I feel like that's why inflammation is invariably associated with hypertension and vice versa. But hypertension will exacerbate that turbulent flow in a way that it imposes a large amount of force in those areas. And over time, you'll get plaque accumulating in those areas and actual atherosclerotic lesions in those areas that results in plaque, as opposed to the areas that experience the most shear stress, the ones that have just the concentrated force of blood hitting those areas. It's very ironic, but that is the explanation. I mean, it's the best explanation we have for that phenomenon, but we do know that in practice, it is predictable where these set sites will develop. It is in the areas that experience the most turbulent blood flow, and it is exacerbated by hypertension. So is shear stress, but it doesn't seem to be enough to cause large amounts of plaque, at the very least large amounts of plaque, if any, in the areas that experience the most shear stress. So you can imagine the aortic arch.
Yeah.
You would think that the shear stress happens at the U part. It's an inverted U shape. It happens at the base of the U, you can think. So at the top, basically, in this case, because it's upside down. That's what experiences the most shear stress. But you'll actually see atherosclerotic lesions occur on the undersides instead of the top, because that experiences the most turbulent blood flow whenever it's pushed through. So that's what causes these. That's where the plaque builds up the most, and that's why we say, well, cholesterol obviously has a role to play in heart disease because it's involved in cellular repair anywhere. But if it were the absolute cause, then you'd see it develop everywhere, and you'd also see it theoretically develop in equal amounts at each time as well. Not just everywhere, but equal amounts. More cholesterol, you've got, more buildup everywhere as well, in the venous side and the arterial side. So anyway, I hope that clears it up. If it didn't, you can ask if you need any more clarification about that. Did I cover everything?
No, I think this definitely helped a lot more.
Okay, good. Just wanted to make sure. Because I know that sometimes I get lost in the weeds because my brain goes off in 1,000 different directions all at once, and then I'm like, I have to. I get lost in the weeds, and I have to remember my way home, kind of thing. That's the best way of explaining it. But yeah. All right.
I think that covers everything that I was curious about, at least as of now. Who knows? I might get some more questions later on.
Well, if you do, since you are a member of the program, you can definitely schedule another one because that's part of my job, being affiliated with this, and I do enjoy. I don't want you to think. I only look at this as a job. I like these conversations because also sometimes my brain gets a little rusty. It's not challenged enough. And so then when someone asks me a question, I go, oh, okay, let's go. Let's respark that. Let's rekindle that part in my brain. I actually just had that. Yeah. I remember I actually just had that happen to me earlier when I was recording a video about like digestion or something. It was lipoprotein metabolism. And I went, oh, and I stopped recording and I reread my notes and I was like, man, it sometimes is a little discouraging, but no, it's good. It's healthy. It's healthy. So anyway, yeah, if you once again, if you have any more questions, you can reach out to Peter or me directly, actually, and I'll talk to Peter about it, and then we'll schedule another one. So yeah, this was good. Hope you learned something.
Me,
yeah, definitely, no problem.